New insights and technological advances in disease biology are yielding new classes of novel medicines with dramatic improvements for patients and opportunities for curative potential. In this session, emerging industry leaders discuss the progress made across a spectrum of new modalities and critical issues to overcome to achieve success. Please welcome Deborah Barton, CMO of Charisma Therapeutics, John Houston, President and CEO of Arvinas, Eric Kelsick, founder and CEO of Dino Therapeutics, Devin Smith, CEO of Arbor Biotechnologies, and session leader Ryan Kaywood, CSO of WuXi Advanced Therapies and Oxgene.
Okay, so welcome to the panel. This is the panel on next-gen new modalities. It's a suitably broad name for the panel, reflecting really the armory of different technologies that we have now to be able to improve patients' lives. So it's my pleasure to be able to welcome to the panel some esteemed colleagues, starting with Deborah Barton, who is the Chief Medical Officer at Charisma Therapeutics. Charisma are developing cell-based therapies using macrophages, primarily for oncology applications. We have Devin Smith, who joins us as the CEO from Arbor Biotechnologies. So Devin is developing entirely new CRISPR platforms for the treatment of genetic disease. We have John Houston, who is the President and CEO of Arvinas, and Arvinas are really showing everybody what you can do with small molecules in terms of developing bispecific small molecules that allow you to degrade specific proteins. And then we have Eric Kelsick, who is joining us from Dino Therapeutics, who is the CEO and founder, who is developing new AAV technologies for improved delivery and reduced immunogenicity. So it's really great to have you all on the panel. Thank you very much for joining us. I thought I'd just start by just setting the scene. So we're 10 years on from when the WuXi Global Forum first started. And I think it's important that if we all just take a step back and say, okay, well, what was around 10 years ago? So we didn't have CRISPR. There was only one patient that had been treated with CAR T therapy. There was very little efficacy in the AAV field. Gene therapy was really, in the doldrums at that point. So it's an incredible time for us all to be a part of this journey. And it's really great to have you all here on this platform. So I'd like to start by talking maybe initially about cell therapy. So as a field, it's exploded massively. And Deborah, you're very much involved in cell therapy. Where do you guys see the field heading?
Well, thank you, Ryan, and good morning, good afternoon to wherever you are in the world. I think cell therapy has been evolving significantly. As you said, 10 years ago, we were not even there. And now CAR-Ts have been extremely successful in hematological malignancies. But if I can bring the conversation a little bit to solid tumors pose a number of challenges for those cell therapies to act. And they do require a different approach that needs to be developed. So trafficking of these cells, if you imagine a CAR-T cell getting into the solid tumor is really physically difficult, versus when you compare with treating flowing leukemia cells in the blood, for example. Yeah. So the solid tumor microenvironment it removes the T cells while it brings the macrophages in. So it does make sense to explore CAR macrophages for solid tumor. There's a lot of innovation that needs to be brought in place to tackle the other diseases that are not currently being addressed by the CAR-Ts that are growing so successfully. I think, especially for solid tumors, delivery is one of the biggest obstacles. So again, for blood cancer is a whole different thing, but CAR macrophages, they have an advantage because macrophages are already present in the solid tumor. So they're the most abundant cells in there, and they are able to phagocytose, kill the tumor cells, and also process and present the whole variety of neoantigens, because we know solid tumors are different than leukemias. Leukemias are homogeneously expressed, for example, CD19, while solid tumors, each cell can really express different things. So access to the solid tumor is one of the biggest issues, but also the individuality of the solid tumor is something that needs to be addressed.
And so, Devin, you're primarily working on gene editing as a principle, but are you using that in a cell therapy context, or are you using it in a viral vector context? So what's your main application?
Yeah, so we actually, you're right, there's two ways we can utilize gene editing. We can utilize it in a cell therapy context or in a genomic medicines context. So on the cell therapy side, we're actually partnering with folks who have the key technologies and capabilities there. So we did announce a partnership with Vertex in August in the cell therapy space, where they're utilizing our technology across 7 of their diseases, 3 of which were noted: sickle cell disease, beta thalassemia, and type 1 diabetes. And so I think it's really fun to watch the expansion of cell therapy beyond liquid tumors. Now we've got solid tumors like Deborah's approaching, and then you've got these other approaches in other diseases that I think it's fabulous to see this expansion. So for us, it's about partnering on the cell therapy side, and we have that one announced, and hopefully several others coming along.
And if I may add, because we're also looking at gene editing, as you know, for macrophages. The checkpoint is CD47 SERP alpha. So we are also working in CRISPR editing the CAR macrophages to knock out SERP alpha. And we have some very interesting preclinical results. So there are many ways to always keep improving and keep looking for other things that we can do with the CAR-Ms, and gene editing is definitely something that we're seriously looking at.
And when you're saying you're using gene editing, you're using it as a mechanism to knock out genes that are stopping the cells from being active. Is that a fair comment? Because that's a similar issue that you have with T cells, right? They become allergic, they don't like the environment, they turn off. So it sounds like that's what you're mainly using it for. That's
exactly what we're doing. We're currently moving from in vitro to in vivo studies, and hopefully that will keep progressing quickly.
Okay. And you're in Charisma, you're in the clinic, is that right?
We are in the clinic. So we have a phase 1 first-in-human clinical trial. We're enrolling patients in 4 sites: UPenn, UNC, City of Hope, and MD Anderson. And we're studying CT-0508. It's a HER2-targeted M1 polarized CAR macrophage. We're enrolling all patients, not only breast and gastroesophageal, but any patient that expresses HER2. We treated 4 patients today, so it's very exciting. Yep. We haven't observed any dose-limiting toxicities. And importantly, we're collecting pre- and post-treatment biopsies to look at this tumor microenvironment activity. And single-cell RNA sequencing is showing in the patients the remodulation of the tumor microenvironment. So it is showing the signals that we were expecting. The data was presented oral at SITC last month.
Brilliant. So, I mean, cells are obviously highly motile, right? So they can respond to their environment, they can find their way into different tissues. I mean, John, in your case, obviously you're using small molecules. I mean, I'm assuming the fact that the small molecules aren't motile still gives, but the fact that your molecules are so small gives you a major delivery advantage. Is that fair?
Yeah, PROTACs, which is the major chemical that we design, proteolysis targeting chimeras, are small molecules. They're larger than average small molecules, more in the range of 800 to just over 1,000 Daltons. So they don't naturally lend themselves to either being orally bioavailable or brain penetrant. But since Arvinas was founded in 2013, we built in the design features that truly make these molecules orally bioavailable with systemic delivery, just as an oral pill. And we also designed them as brain penetrant. So we have obviously a very nice growing oncology portfolio, but equally excitingly, a growing neuroscience portfolio.
Okay.
Sorry?
That's interesting. That is interesting because, yeah, clearly the blood-brain barrier is often a major issue in terms of getting larger macromolecules into the brain. So, yeah, that's amazing that you've been able to do that.
Correct. And it's one of the exciting things about a platform where there's a novel approach. I mean, the first paper on PROTACs came out 20 years ago. It was Professor Craig Cruz, Ray Deshays, and they talked about this way of designing a molecule, a heterobifunctional molecule that could hijack the cell's natural proteasome and ligase machinery, and basically target a specific protein, drag it into proximity to the ligase machinery, have the ligases tag the protein with ubiquitin, and then it get dragged off to the proteasome, which is basically the garbage disposal unit for proteins in your cells. It's a great proof of concept paper. And then in 2013, the company was founded on the back of that with Craig Drews as the founder. And we've been working on all the different design features of the platform since then.
Oh, that's amazing. Is that, I mean, well, Eric, I'll ask you about the one in the middle in a second, the AAV that sits between the small molecule and the cell. But so, John, I suppose a major issue that we often have with small molecules is we might be able to make them bind, but actually making them stop the protein from functioning is obviously a challenge. You're getting it to fit in an active cleft that stops that protein from working. But it sounds like your solution doesn't really matter as long as it binds to the protein because it just drags it for degradation. Is that a fair assessment of the approach?
Exactly. Most inhibitors are occupancy-driven events where they sit on a receptor or an active site in the enzyme. Now, PROTACs can certainly bind to those active sites, but they can also bind anywhere on the protein. Yeah. And what you're really doing is allowing this protein-protein interaction to occur between the E3 ligase and the protein. And once that E3 ligase is in proximity to the protein, you get ubiquitination. And once you get 4 ubiquitins transferred to lysine on the target protein, that serves as the signal to send it off to the proteasome. So you're getting rid of the protein completely.
Cool. Very, very cool. Yeah, really different. I like it. And so, Eric, yeah, we sit with AAV somewhere in between the cell that can move and the molecule that has no biology in it. So tell us about the issues with AAV. I mean, delivery again, you know, how does what you're working on solve it? What are the major bottlenecks and issues with gene therapy, particularly systemically?
I'm happy to touch on all those. Maybe to step back and say, by way of introduction, what's the mission at Dino? We have a saying for our team. It's like a mission statement, empowering a diverse team of the best problem solvers to drive cutting edge science towards improving patient health. So we always want to be at the cutting edge, and that's how we began. We saw this opportunity with gene therapy, which is a really exciting new modality. And there's some interesting differences about gene therapies that mean we can apply technology in new ways to have greater patient impact. An important difference is that gene therapies, they always have 2 components. There's the vector and there's the payload. And they're quite modular. You can put many payloads into the same vector. And so Dino's approach is to focus on solving the vector portion because the delivery challenge has always been, you know, the biggest in gene therapy. It's what's been limiting progress in terms of the number of diseases we can treat and the patients that we can treat with those therapies. We took this approach because there's new technologies coming online now that really help us to engineer proteins in a fundamentally new way. And by focusing on solving this problem, the modular part of the gene therapy, Dino is working to maximize our impact on patients, in particular through a partnering business model in which we work with other world experts in gene therapy who provide a payload as well as the commercialization and development pipelines to bring more products to patients. So the underlying technologies that make that possible, at Dino, we have an AI-powered platform. My background is actually scientific. I led a team at Harvard starting in 2015 to develop some new capabilities that enable us to engineer proteins in a new way. And then Dino was founded in 2018. We've been operating in Cambridge ever since.
And so in terms of the work you're doing, it sounds like you're working with a diverse number of different therapeutics companies in the field. I assume the challenge you're facing with gene therapy varies with the disease, the route of delivery. You know, are you evading the immune system or are you improving circulation kinetics? I mean, is that a fair assessment of the sort of work that you guys are working on?
That's exactly right. And that's part of the opportunity of why it makes sense to focus on enabling technologies, because every company that's building gene therapies really has the same wish list in terms of what an ideal vector would be. It would be extremely efficient to the target tissues, but also very specific, only going to those cells and not going to other cells or off-target organs. It would avoid the immune system, especially pre-existing immunity, which means that because many people have immunity to natural viral vectors, actually they're ineligible or wouldn't be helped by the gene therapies that are under development today, which is really not acceptable as a field. We want to be able to help every patient who has the disease, not just a subset. There's the packaging size, and then there's the manufacturability. So it's been a challenge to engineer AAVs in the past, or really any means of getting to those cells. AAV vectors are just the best thing that we found in nature, and they come with them limitations because they never evolved naturally to, for example, go into the brain, cross the blood-brain barrier. There's no reason to do that. However, the protein itself is very complex, and proteins in general, we don't understand all the fundamental biochemistry of how they work and how they fold, although there's a lot of exciting progress being made and going past that lack of understanding to be able to actually design proteins from structure alone. But it's also a challenge in AAV because the protein itself is so complex. It's 60 different monomers that come together to form the shell. So just one tier above what would happen for a single protein, as well as there's a lot of the basic biology that we don't understand, in terms of how AAV interacts with human body, the receptors, the way in which tropism is determined. Actually, as one of the interesting vignettes from the work we did at Harvard, we actually discovered a fourth gene within the AAV genome that has a lot of questions about how it functions, but there were only 3 known to date, despite that we've studied AAV.
Is this the
50 years.
MAAP? Was that the one?
Yeah, it's membrane-associated accessory protein, MAAP.
Yeah.
So that just came out of, our approach to engineering the protein was to take a very systematic kind of scan across every position, mutating to every possible amino acid, and that was designed to teach us how to engineer AAVs better. But as a consequence, we also noticed these patterns that made us realize there's something surprising happening here. We looked into that. I guess to bring it back to what's the challenge that we're trying to solve with protein engineering, we, in the past, there's been 2 canonical approaches, directed evolution or rational design. Directed evolution is like a lottery ticket approach. You're trying to maximize your chance of winning by buying a lot of tickets, even though for any one ticket, probability is very low. Whereas rational design is about, using the mechanism, our intuition, to make better guesses. And the challenge with AAV being that we just don't have a lot of information for how to do that. So the opportunity that we saw was to do this in a more data-driven way. We now have capabilities of measuring many, many variants through high-throughput DNA synthesis, high-throughput DNA sequencing. You can combine this together in what's called a DNA multiplexing experiment. And what's most exciting is you can actually do these experiments, measure hundreds of thousands of variants in animal models, which is very relevant data for predicting translation to humans. So we developed that capability in the lab, and that was published in Science. And then there was a second challenge that created, though, which is how do you analyze all this data? And to solve that problem, we also automated the analysis and design of new experiments using that data through a machine learning-based approach, which is also published in Nature Biotech. So those capabilities is what really fundamentally enables us as Dino to be a specialist in solving this in vivo delivery challenge, which is the value proposition we have for all of our partners.
Cool, thanks. And I think we'll turn certainly to talking about the impact of structural biology and bioinformatics, shortly as we go through. I mean, I think you mentioned an important point about pre-existing immunity, obviously a well-known problem in viral vector space. I mean, John, I had a colleague who works in the US who once told me he had a vaccine and he could vaccinate me against caffeine. I didn't realize you could actually get antibodies against small molecules. I assume that's not a problem that you see with any of your molecules, saying they're slightly larger. Is that potentially an issue?
No, there's no haptenization or anything occurring. There's no immune response to these small molecules at all. As I say, they are small molecules, so they act and perform just as normal small molecules. Good systemic delivery everywhere, and there's no antibody response, which is great.
And so Deborah and Devin, on the cell therapy side, obviously moving from autologous to allogeneic platforms, I mean, is that a pipe dream, do we think, or do we think that transition will eventually happen?
Yeah, I think the allo platforms, they are coming up, and this is at Charisma is something that we are actively looking at, and this is one of our active areas of discussion in potential collaborations. And it will have a significant role in decreasing costs. I think we're going to talk about costs later. Yeah. But allo is something that we are looking at in the future.
Yeah, I mean, I think clearly, as you say, we can move on to cost in a little while, but it would transform the cost base for cell therapies. It really would. Devin, anything to add on that?
I think that you're seeing it beyond, I think, the CAR space, all the different oncology approaches. I think you're seeing that quite a bit now. In Vertex had published one patient data on an allogeneic approach for type 1 diabetes. I think you're starting to see more cell therapy allogeneic approaches coming out outside of oncology, which is also quite exciting because, I think the opportunity space there to revolutionize is really there. And I think having the right tools to do so that allow us to do it quickly and efficiently is important.
Cool. And so Devin, on the gene editing side, so I mean, you're in a sea of Cas9s, right? When we look around, there's many companies working on Cas9, there's different platforms, and you're going in a different direction, discovering entirely new platforms. What's the rationale behind that, and what's the main driver? Is it solving an industry bottleneck that you've identified at Arbor?
Yeah, it's a great question. There's a couple of reasons. So one, remember, Cas9 wasn't the best nuclease found; it was the first one that worked. And
Like
right,
well.
it's the one that works, so that's the one people ran with, which is often how science occurs. But our co-founders, Fong Zhang and David Walt, got together and said, well, why don't we actually go back to the metagenome and identify novel, differentiated nuclease technology that allow us to do more than we can currently do with just Cas9. So with Cas9, you've got a specific PAM or binding site that limits you to what parts of the DNA you can actually impact. And so over the last couple of years, we've built a discovery platform where we've been able to identify a portfolio of nucleases that'll give us a diversity of PAMs, a diversity of cut types, and a diversity of size. So we have much smaller nucleases that can easily fit within an AAV if we choose to deliver via AAV to areas outside the liver. So by having this portfolio approach, we flip it on its head and we actually start with the disease biology. Say, what is that underlying pathology we need to fix? And then let's bring the right editing technology to bear against that rather than have one hammer and everything's a nail approach. And so it's really, I think, taking a page from a more traditional small molecule view of the world or an antibody view of the world, which is we have a platform, let's figure out what we need to fix first and then bring the right tools to bear against it. And then we can do that in vivo editing appropriately. And for us, we're targeting the liver and CNS. Liver, obviously, we're going to deliver via lipid nanoparticles. CNS, there's a variety of approaches. I think Eric mentioned AAV is obviously one interesting approach, and there's other non-viral approaches that folks are looking at on delivery as well. So it's an exciting space, I think, with the opportunity to do more than just knockdowns now on the editing. We can do precise edits. We can knock in larger cargo. So it's a fantastic and rapidly emerging field.
Yeah, very cool. Is anybody else, in terms of either on the research side or other work that you've done, had much interaction with the gene editing side of things? I mean, Deborah, you mentioned that you'd use it slightly. Eric, is it anything that comes up in the work that you're doing on the screening side?
Well, I think about gene editing just as one category of gene therapy. When it comes to Dino, like whatever partners want to do on the payload side, anything is fine. And we're just there to help them make their therapies better by solving this one specific challenge of delivery. And maybe in the future, we'll expand to other aspects of the platform, but we always want to be helping our partners and not competing with our partners. That's really core to Dino's model of trying to maximize their impact on patients.
Yeah, and so, I mean, if we just think, so John, Deborah, Devin, you're all going clinical with your products. That's correct, right? And whereas Eric, you're mainly partnering with companies such as Deborah's, John's, and Devin's. That's correct, yeah. So, cool. Okay. So, I mean, one thing that's changed a massive amount certainly, I mean, when I first started my PhD, I had to handwrite primers and scan them off to Sigma, which was not that long ago. So, now we can order thousands of DNA, base pairs from a number of different providers. You can access genome of the white shark if you want to. I mean, how has structural biology, bioinformatics, access to data, I mean, how has that impacted all of your development programs? I mean, open question to anybody that wants to pitch in first.
So John, certainly AI in general, I think has been very impactful. We're seeing in our early discovery part of the pipeline, more use of structural biology, AI. We do use CRISPR as a tool to help validate some of the targets. So the whole panoply of different technologies and tools that you can deploy have been getting better and better and more accessible, even for very small companies. So that's been exciting. In the old days when I was in big pharma, these were big, expensive areas. Really only big pharma could deploy them. And now you're seeing accessibility go to these smaller companies. So it's hugely enabling.
Eric, you were talking about, I think I read the work, I think maybe early this year or late last year, I can't remember, about his algorithms where you can actually now predict the folding of proteins, of individual proteins. I mean, is that something, clearly that's a part of the workflow that you're developing?
Well, I would say it's an inspiration. It shows you how powerful the new machine learning capabilities are. especially as we begin to adapt them to speak protein language rather than, say, speech and images, which is where they were developed from tech. I would say it's inspiring because the way I think about the problem that we're solving in Dino is we just want to make the best possible protein, the best possible vector. So we really need to predict function. And it's quite exciting that we can now predict structure from sequence. But what we really want to know is, for a function, what is the sequence that will provide that function? So there's a question of, do you actually need structure? Do you want to go from sequence to structure to function? Because now we can do the structure part, sequence to structure, but going from structure to function is still an open challenge. And the way we're approaching in Dino is, we think it's possible to build these really powerful models that are predictive using machine learning, but we're actually directly mapping between the sequences and the function themselves. And maybe the model is going to learn to do that by learning something about the structure, but there's also a lot of other things that we might be learning. We don't really care exactly how, so long as we're able to build predictions and then to quickly validate them, which is also something that we do experimentally. So I would say it's inspiring the new capabilities, but we're more directly applying those towards impact for solving, transformative problems in gene therapy.
Yeah, I think the words you're using there, I mean, I would have never thought that an algorithm could have predicted the complexity of protein folding until I saw the news and I realized it could. I was utterly blown away. And Devin, from what you were saying earlier, it sounds like you're mining sequence on a daily basis. I mean, are you also going out into Arctic vents and things like this and isolating new species that were previously unknown and then sequencing them? Is that part of the plan?
So there certainly is a part of what we do. The other piece, I think, is we've got a database now of over 3 billion proteins. So we actually have a computational group that actually is a bunch of ex-Google algorithm data mining experts that can go through this and identify whatever it is we're looking for, motifs of interest, and then we pull those into some high throughput screening approaches. And AI and machine learning are critical to what we do as well, because once we pull out systems of interest, we have to run them through an engineering platform to engineer them up to get their functionality high in the million cells for their nuclease functionality. The other piece of that is, by applying structural biology approaches, we can then do exactly what Eric was talking about, which is you've got sequence, you then have structure, and you move to function, so that we can then begin to tie specific mutations we make within a nuclease to functional benefit or functional readouts. And so that utilizing some of these more predictive approaches on top of structural biology is very enabling for us. And so it is an important part of what we do. And it's exciting now to have so many tools that allow us to computationally predict things rather than spend months trying to figure out if it's an approach forward in the wet lab, for example.
Yeah, it certainly seems incredible. And, as somebody who's spent most of his time in bioinformatics is simply doing a blast alignment, it's incredible what these different platforms and technologies can do. So, I mean, we're talking about advanced therapies. We've, as a species and as an industry of medicine, we've gone from small molecules, we've gone to antibodies, and now we're going into even more complicated vectors and cells. These things are also getting quite expensive when we go along. So, I mean, where do we see the major cost savings coming from? What can we do as an industry to try and decrease the costs of some of these molecules?
I think when you look at the development of a platform or a technology that's going to be delivering a therapeutic, the cost of those materials is a significant factor. The scalability of your platform is key. We interacted with a lot of small companies with platforms, and usually we found them ending up in a niche setting. So that's always the challenge with a new platform or a new focus. How do you move away from niche validation in small subsets of patients potentially, and broadly have it deployed against a whole range of diseases and have it deployable against a whole range of different patients? We've got an advantage because it's a novel technology, protein degradation, but it's a small molecule. So we won't have those issues in terms of scaling, and we can work in multiple different diseases. But I do think that is one of the challenges for new platforms: the costs of production, scalability, and broad spectrum nature of your platform.
Yeah, and certainly scalability is an issue we've seen in the viral vector space, and particularly as there's been a sort of a, everybody wants to get into the clinic, particularly cell therapy as well. Is part of what Dyno's doing focusing on trying to ensure that these vectors are more easy to manufacture or being produced at higher yields?
Yeah, absolutely. And I can actually give two answers to that. Because there's, as I said, there's this wish list of properties, including the efficiency, including the manufacturability, that are important to everyone doing clinical gene therapy. And it's important to optimize across multiple of them. Like, you don't want to get better at efficiency, but then worse at manufacturing. So one of the things that the new technologies enable us to do is to optimize across multiple properties, because we're directly measuring each of them, not to accept, getting worse in one dimension and only getting better in the most relevant dimensions. But by improving delivery, we directly solve many problems. We both make the therapies more effective, make it possible to treat patients by going into more cells. We also make them safer because we can target them specifically to those cells. And if we further improve the efficiency, now there's also the potential to effectively treat the disease, but at lower dose. And because now you can make more doses per batch, you can treat more patients and get through the manufacturing bottleneck. So all these things are really, fundamental to the progress of gene therapy as we begin to realize the potential. Right now, there's just 2 FDA-approved gene therapies. And so we're really just at the beginning when all the pieces have come together, and there's many, probably generations of gene therapies that will come out, each making advances in these different dimensions. And that's what convinced us at Dyno to commit to this, approach, which is designed to maximize patient impact, but we do that by being partner-centric and working with many partners across all the areas that could benefit from improved delivery.
And I think there's a general feeling in the field that obviously that viruses are the most, or one of the most efficient ways to deliver genetic material. But when I think I first learned that polio, only 1 in 10,000 of the particles is actually capable of infecting a cell. You think, okay, if you were using that for gene therapy, that would be a pretty poor vector. And similarly with AAV, I mean, we often see that batches are, only 2 or 3% are actually able to get into the cells and transduce them. So, as you alluded to, if we could improve that and just ensure that the particles that we're delivering to patients are not stimulating the immune response but are actually going to cells, then clearly that would have a massive benefit. I mean, Deborah and Devin, so on the cell therapy side, I mean, it's a very local treatment for most instances at the moment. So it's bedside treatment in many cases, and sometimes people are establishing facilities for cell processing near hospitals so that they can process them there. I mean, is that a major influencing factor in the cost of goods as we see cell therapy at the moment?
Well, before we go there, I just want to connect with the last thing you were talking about, the viral vector. And the viral vector is indeed very important, and it is a very important portion of the cost. And we have developed a proprietary adenoviral vector, and we're able to decrease the cost of the viral vector by 10-fold. So we can do things in-house, and we can figure out ways to be creative and try to find ways out of that. And then there's the cell product, right? So it's an autologous cell product. And as you said, there's a whole infrastructure that goes around it. We're constantly looking into decreasing the vein-to-vein time and increasing the cell yield. So for example, one of the efforts we're looking at is utilizing CAR monocytes instead of CAR macrophages.
Okay.
Monocytes are the precursors of macrophages. The cell yield of a manufacturing run is much higher, and the manufacturing time is much shorter. It goes from 7 days to 1 day. So even in the autologous cell therapy field, we can work and try to find solutions to decrease that cost.
Okay, and then Devin, on your side, I mean, Devin, are you at the clinic yet, or are you still in the sort of research and development phases at the moment?
Yeah, we're still a preclinical company, but given we sort of partner on the cell therapy side and then we are in the genetic medicine side, I think we see both. I do think there is a large opportunity to decrease cost of goods. I think allogeneic cells will help. I think scale helps. I think on the genetic medicine side, where we're doing in vivo delivery via LNPs of editing technology, I think the biggest help there on the cost of goods on LNPs, obviously, it's one of the few benefits I think of the whole COVID mess is, people now know how to make LNPs and all the RNAs that go into them at scale relatively inexpensively. And so that's really benefited us from that perspective. Well, I think, when we move to other delivery modalities like AAV for some of our, for our payloads, I think that's where, some of the work that Eric and others are doing is going to be critical. But I think we still have a ways to go, but I think folks recognize the importance of it. And I think there's a lot of efforts going on. I think the other piece that you've seen a shift over the last few years is starting CMC much earlier than we used to. I think 10 years ago or 5 years ago, people would worry about CMC when they were ready to file their IND. And the problem was that's what's killed us in this cell and gene therapy space. And so I think now people worry about it much earlier.
To the public opinion side, in our field, we're talking about treating patients quite often with highly complex vectors, systems that are alien to patients and public quite often. I mean, how do we keep the patients and public opinion on side of the different platforms that we're developing?
There's a question of how are we talking about our products and how do we make sure that what we're doing is really understood? Like in the case of viral vectors, actually, I prefer to use a different term. I just prefer to talk about capsids themselves. Capsids are a protein shell. It's a technology which we've learned about from nature, but we actually take out all the viral parts and now we use it to deliver a therapeutic DNA. And who are we helping with that? We're helping a patient who has a genetic disease. And for most of the genetic diseases, because small molecules and antibodies, for the most part, are inhibiting their targets, what's needed to rescue these phenotypes is actually adding new functions, adding back in the protein that's missing. So for that reason, most genetic diseases don't have good treatment options. And this is, a very promising way in which we can help those patients where there's not many alternatives. And I think making that need clear that we're really doing things which are going to help a huge number of people. And then, being thoughtful about how we explain it so that what we're really doing is clear.
Deborah, anything that you I don't know what work you've done at Charisma on the public engagement side, but anything you can add there?
Yeah, I completely agree with having the value very clearly stated when you know the benefit. And currently I'm in clinical trials, but when you know that an oncology product prolongs life, maintains quality of life, those are the things that need to be emphasized to create value for the patient, for the communities, but also even before that. So we had recently a call with a patient advocacy group and a group of patients, which was amazing. And most of their questions were about clinical trials, clinical trial design. How does it work? How do you ensure our safety or giving something new? So there's so much education. Sometimes we overestimate what people know about what we do. So I think there's a lot of education that needs to be done from the clinical trials all the way to the explaining of those complex therapies. The more, the better, and the simpler, the better.
That's great. So I think we're close to time now, and I'd just like to first say thank you, all for your incredible input into the panel. This is 10 years since the WuXi Global Forum first started, and 10 years, as I alluded to at the start, since all of these incredible innovations that have powered and fueled the industry. What do you see in the future? Just a very quick prediction from each of you for what will happen over the next 10 years. Just throw it out there, anything, and then we'll have a call in 10 years and we'll see how we were all wrong. So John, first, any predictions for the future?
Well, it's more of a hope, and I do. First of all, I hope all the technology platforms being discussed today come to fruition in terms of clinical benefit. That's an absolute, because that's really what we're focused on. Particularly, I hope that there's major breakthroughs in the neuroscience and disease area. That's a huge unmet need. And I hope in 10 years, we'll be talking about breakthroughs in diseases such as Alzheimer's and Parkinson's. More of a hope, but we're a lot of companies on that path to making that happen.
I think we all hope that, yeah, we agree with you on that one. So Deborah, prediction for the next 10 years.
Well, my prediction is, I want to watch it in 10 years, is that we'll be able to develop a really patient-centric, customized therapy, which will include several aspects of the immune system, probably a number of immune cells, not one cell, but an agreement and a coordination, targeting a number of different tumor antigens, not only one. So I don't think one cell against one antigen will resolve the solid tumor issue. So I think it's going to be something orchestrated and very much tumor-centric, patient-centric.
Excellent, interesting. I wouldn't have seen that coming. So yeah, let's watch that space. Eric, from you, next 10 years, what do you see?
Yeah, well, two predictions. One is we've talked about the amazing progress in gene therapy to date, and I think we're at the cusp of something really important. You know, in 10 years, I think we will solve the in vivo delivery challenge. Thanks to the progress that we've made, we now have the pieces, and thanks to these new tools that make it possible to engineer really transformative improvements into the proteins themselves. So what that means is going into every organ, every cell type very precisely through some means of delivery and at doses that are thousands of fold lower than what are being used in the clinic today. That's the opportunity, I think, on the gene therapy side. And then the prediction for some of the other technologies we talked about, in particular, the AI and machine learning technologies. We've really seen in the last few years the rise of biology platforms and understanding from the entire ecosystem, from partners to investors, about how they work. And I think that what will happen in the future is similar to we've seen a consolidation of pharma along the dimensions of commercialization. We'll also see a consolidation along the lines of these fundamental technologies, in particular, aggregating data. I think the companies that have the most data will be really dominant when it comes to creating new therapeutics.
Excellent. Thanks, Eric. And then Devon, finally from you, prediction for the future.
I think I like what everyone has said. I think fundamentally in 10 years, I'd love to see a raft of novel therapies out there that change patients' lives. And I think it'll be a mix of cell therapies, some genetic, whether it's a gene therapy or other genetic medicines, and some small molecules, because I think the opportunity is still tremendous. I think John's spot on. Hopefully this is the decade of neuroscience. The last one was supposed to be, but hopefully this one will be the one that actually translate those to therapies. And so yeah, my hope is we see a lot of new therapies out that shift to curative or cure-like efficacy for patients, because that's why we're doing this.
Excellent, thank you. My prediction is that in 10 years, I still won't be able to use bioinformatics, but let's hope that I prove myself wrong. So yeah, John, Deborah, Eric, and Devon, thank you so much for your time. It's been an absolute pleasure having you on the panel, and I hope that we get the chance to catch up again soon.